welcome to those of you we can't see in the room, and certainly welcome to all of the faces that I can see in the room. We're happy to have you here. My name is Danny Fallon. I'm the director of the Wendy Clegg Center for Autism and Developmental Disabilities here at the Bloomberg School of Public Health. We're thrilled to have this symposium today for many reasons. The first is that this is part of the centennial year of the Bloomberg School, and so there are activities throughout the entire academic year celebrating 100 years of excellence in public health and thinking about what comes next in the new century of public health. We are not quite that old as a center. We are only a little over 2 years old, and so 2 years ago, almost to this week, we had our first symposium for the center, and so we're thrilled that we were able to bring together another esteemed panel of folks to have our second symposium, and then we're trying something new this year after the symposium, which should end at about 4:30 today, maybe a little bit sooner than 4:30 today. We will have a poster session downstairs in Finestone Hall. Actually, no, we're on the second floor, right? So around the way in Finestone Hall, which has about 45-ish, 50 posters. So I think there'll be a lot for everyone to see, and you'll get to hear about what's going on with some of the research of students and faculty here, and also folks from other institutions nearby. So I'm really looking forward to that, and I hope that many of you, if you're popping in and out of this, can then come back and join us for that poster session and reception. So the Wendy Clegg Center, as I said, was only started 2 years ago, and it's named after the late wife of our dean, Mike Clegg. Wendy was a strong advocate for children and for child's health, and particularly for developmental disabilities. She and Mike have a child on the spectrum who is now an adult on the spectrum, and they have been quite dedicated to the impact that public health can make on the problems and solutions to autism spectrum disorder. And so we are incredibly grateful to all of the folks that have contributed in Wendy's honor and to the center for its mission to make an impact through the lens of public health. And so today I'm very excited to introduce our keynote speaker, who is Dr. Wendy Chung, and she is the clinical research director for SFARI, which some of you may not know that acronym. It's the Simons Foundation's Autism Research Initiative. And she not only directs that, but has led an initiative that I hope she will tell us about today quite a bit, so I will leave that to her. She's also at Columbia University, where she is the director of the clinical research there, or clinical genetics, sorry, clinical genetics there, and has been a longtime colleague and friend to many folks in the room, and we hope that we get to see more of her as well. So welcome to Wendy. Advanced, but I'm wrong about that. Right there it is. There you go.
Okay. Thanks so much, Danny, for having me here today. I'm really honored and privileged to be able to spend the afternoon with you. And this is meant to be very informal, so if I say something that's not really making sense, tell me to back up the bus and I'll be glad to stop. And if I started going away too fast, just tell me to slow it down. So I am going to talk a lot about, this is an unabashed advertisement, I'll be forewarned, to be able to tell you about some of the initiatives that we have at the Simons Foundation, with the hope actually that many of you are actually going to use those resources. So this is hopefully meant to be an infomercial in part in terms of what we're doing. So I'm going to breeze through a bunch of this. I'm going to divide this talk really into 3 different sections, a little bit to give some background in terms of the genetics of autism, a little bit more to tell specifically about one initiative that we've done, taking a deep dive in a genetics approach in autism, and then finally to talk about this new initiative that we have that we call SPARK, which is actually a national collaborative or national autism cohort that we're going to be doing that's going to be hopefully 50,000 individuals and their families, so very large scale in terms of this. So the point, which is really what along with the symposium, is that one size doesn't fit all in terms of autism. And these are literally the faces of many lovely young individuals with autism. And as you can see, there's really a range in terms of disabilities. And I think that's one of the greatest challenges for all of us when we both think about this, as well as when we try and address the needs of individuals with autism, is it's not just individuals who have severe impairments, and it's not just individuals who have mild impairments. And sometimes the conversation, in fact, gets a little bit confused because we try and cover everyone within one term, but actually it's quite diverse. And so as we're doing that, one of the things that I'm going to try and make the point of is that autism, to me, in many ways, is so beautifully complex that we're really talking about multiple dimensions in terms of behavior, in terms of how the brain works. And in many ways, we have to be able to put people in buckets. So we have to, say that they meet diagnostic criteria by some dimension that constantly keeps changing, by the way. But at some point in some freeze frame in time, we say they meet those dimensions. But in other ways, what I'm going to try and make the argument about is that this is really just variation that we see in the general population and across many different axes, across many different dimensions. It's somewhat arbitrary when we make a cutoff and say at this particular point, we're going to give you a diagnosis. Within this, I think one of the things that's really remarkable to me as I get to know more and more people, and as I can really see literally how their brains are working in some cases, is how beautifully, amazing and remarkable their brains are. Just because they're not necessarily wired in the same way that you or I, it doesn't mean it's wrong. It's different. And in some ways, they have really unique capabilities, or they see the world in a different way than you or I do, but it's not necessarily wrong or bad. And so I think part of the real beauty of being able to get your head around this is to understand those differences, to celebrate those differences, in many ways to be able to take advantage of those, but yet to support where individuals are really struggling and to be able to help those individuals. So that's a lot about what I think about and as we're trying to do. And so as we think about this, and as we think about, for instance, just as an example, a social dimension to autism, I've drawn this as basically a continuous trait. So rather than saying that, like I said, this is dichotomous or this is yay or nay, you can imagine, for instance, that along the x-axis here might be some quantifiable measure of sociability, someone who's a social butterfly versus someone who's quite shy or withdrawn. And you see a normal distribution, as we do, for instance, for IQ or for other measures. And in many cases, like I said, we will make a cutoff, just like we do, for instance, with diabetes. With diabetes, we officially say if your fasting glucose is greater than 125, boom, you get the diagnosis of diabetes. But if it's 124, are you not diabetic? Are you not anywhere, at risk for getting diabetes? And in the same way, I think many of these things come to behavior as we think about for individuals with autism. So at the Simons Foundation, we've struggled mightily, and we still continue to struggle, and we look for good ideas for all of you that can solve this to figure out what are the causes of autisms. So realizing that this is not one monolithic condition, but there are many different etiologies, and the reason for trying to find the causes are a couple. Number one is obviously if we could prevent it, we'd like to be able to do that in the sense of being able to minimize the burden for individuals. But in other dimensions, and especially this comes to our strategy with the genetics, it's because it's been really, really tough to figure out what some of those sort of things you could hang your hats on were, what some of those really anchors could be in terms of understanding the neurobiology. And so we've used the genetics not because genetics is by any means the only cause of autism or even necessarily the most important cause, but we've had really powerful tools to be able to be absolutely certain that that was the cause in certain cases. And that certainty, that real anchor, that pillar to be able to scientifically be sure of it was why we've invested so much in this historically. But I want to be clear, it's not just all about the genes. And in particular, I think many of us believe, based on epidemiological data, that there are vulnerable periods in terms of both fetal and child's brain development that are especially susceptible in terms of some of these exposures. But being able to put your hands on what exactly it is during, for instance, fetal development, what moms are exposed to, is one of the difficult questions. And I have to say, and many of you know this much better than I, to structure these really good epidemiology experiments, especially in the United States, I have to say, is a challenge. And so some of the answers that we get, unfortunately, have problems in terms of biases of ascertainment or data that we get. And again, I'm just going to put a shout out and an unabashed advertisement. This is for us a very, very important priority to really, truly epidemiologically understand in an unbiased way what these underlying causes are, both genetic, but very importantly, I think we're thinking it's difficult to be able to identify the non-genetic factors, but identify them really robustly and reliably. So if you have good ideas, we've got funding to be able to support them. As we're doing it, though, we are concerned because there are some things that there is good epidemiology. So there are medications, valproate is one of those as an example that moms can be exposed to that certainly can predispose individuals to autism, certain infections in terms of increasing the risk, both again while moms are pregnant as well as the children's brains are developing. And I put a shout out here, I see more women than men in the room. Women seem to always get blamed for everything. So I'm just going to put one plug in while not pointing a finger, that advanced paternal age is actually one of the weaker, but one of the factors that's also associated with an increased risk of autism. So women, take a little load off for a second. One of the things, though, that's very, very clear when you look at the epidemiology is that there's clearly a gender difference, right? I don't think any of us would argue that factor. And so this is not solely a condition of boys or males, but certainly it's enriched in terms of as we look at the distribution out there. And so that's one of the questions we've always wondered, I've always wondered in terms of what causes this, and what is that difference in terms of even normal brain development for men versus women, for boys versus girls? What are some of those differences as we're seeing that? And is this part of that variation of normal? And so in general, when we look at the ratio, it's about 4 to 1 in terms of males versus females across the entire spectrum. But it's interesting that depending on whether you go to the lower end of the spectrum, meaning those individuals with lower IQ, or the individuals who are not quite as impaired at the higher end of the spectrum, that ratio changes. And in fact, once you start getting down to the much lower end of the spectrum, it becomes 2 to 1, or even in some cases close to 1 to 1 in terms of male to female. Whereas when you're going up to the higher end of the spectrum, the higher IQs greater than 120, for instance, then you start seeing even more enrichments of the males versus the females. And so one of the questions is, why do we see those factors? So one of the things that many people thought about, and this is just, I'm sorry, this doesn't project very well, but when you look again by gender, by ages, or by IQs rather, if you look at the females here, for instance, at this lower end of the spectrum, this darker end of the curve here, you can see that the males and females, again, much more evenly distributed as we're seeing that. So as we've been thinking about this, and as Danny was saying, I'm a geneticist, so I think about the world a lot through genetic glasses, although I clearly realize it's not all about your genes. One of the questions is, why should you believe me when I tell you that genes might be involved? What gives me the right to even be up here as a geneticist talking about this? So one of the things we often do is we do these relatively simple experiments of nature, which is we look at twins, right? So twins, they come in 2 different flavors, as many of you know. They come in the flavor of identical twins as well as fraternal twins. Identical twins almost always share the same intrauterine environment and, of course, share the same genes, while fraternal twins could either be the same gender or not, but they share, for the most part, the same intrauterine environment, but may not, share only 50% of their genes in those cases. And so you can look at how close those individuals are in terms of whether or not they have the diagnosis of autism or not. And so one important fact to remember is that for identical twins, there's about a 75% concordance. So that is that if I have one twin with autism, about 75% chance if it's an identical twin that the other twin will have autism. Now, if you're thinking about this, you're saying, well, Wendy, what are you talking about? How can that be genetic? It should be 100%, right? If it's in your genes, and it should be 100%. And that's, in fact, part of the point, right? It's not all your genes. It's certainly not that it's 50% or 20%. It's a lot your genes, but it's not all your genes. And so part of that question is, what's the nurture aspect of this? What are the other, given that the prenatal exposures were the same, what are the other differences that happen postnatally? What are the nurture factors that might be a difference? When you look at those fraternal twins, by contrast, it's only about 25% of those who will be concordant. That is, if one twin has autism, the other twin has autism. Most of those fraternal twins grew up in the same mom, grew up in the same household, grew up in the same environment, so they're sharing a lot of those environmental factors, but that difference, about that 50% difference between fraternal twins and identical twins, tells me there's something in the genes. It's not all in the genes, but there's certainly an important part there. And again, when you look at, for instance, fraternal twins versus siblings, so those individuals that were, shared a different intrauterine environment, likely different postnatal environment, again, you see a little bit more discordance, suggesting that there are some of those other factors besides just those 50% of the genes that they shared. So those are telling us that it's likely that there are some in there. I'm just going to speed through some of these. But when we think about autism versus some other conditions that you might be familiar with, it's clearly not all the genes. And so I don't know how much you can read this, but a lot of people think of cancer as being a very genetic factor. In fact, autism is much more genetic than cancer is. Some conditions that you may or may not be familiar with, depression, for instance, less genetic than autism, autism more genetic. There's a skin condition called psoriasis, though. Psoriasis, in fact, even more genetic than autism is. So it's sort of in the middle in terms of genetic factors that we think about. But one of the things that befuddled us for many years was being able to say that there were some people that developed autism and they had no family history whatsoever. It came out of the blue. And these were a lot of the families, lovely families, who would come to see me. And they'd say, Dr. Wendy, we love being here. You're a really nice lady, but we have no idea why we're here. I have no idea why my doctor sent me to a geneticist. There is no family history of autism in my family. We're doing what we were told, but between you and me, it's a waste of time. And so when we started looking at these, one of the clear factors is that although this sounds a little bit, sort of bizarre, there can be genetic factors that are not inherited. That is, that there can be genetic factors that start brand new with the child, what we call de novo changes, because it's got to happen sometime. Someone's got to be the first person in the family that has a genetic change. And so this is in fact what we saw, and in fact what a lot of the Simon Simplex collection is based upon, is this idea that we wanted to be able to look at mom, dad, the individual child with autism, as well as in many cases an unaffected sibling, to be able to look specifically at the factor by comparing dad's genes, mom's genes, and the individual with autism, see could there be something brand new in that child that might be accountable for that. And so as we did this, we did an inventory of the Simon Simplex collection that is still in fact ongoing right now in the Genome Center in New York City. But as we started this, the resolution that we had to start this about 5 years ago was at the following. So I think of our genomes basically as a set of encyclopedias, 46 of them to be exact. The first thing we could do is basically a counting experiment. See, we're all 46 volumes there. And my kids can do that. They can count to 46, and that was a relatively easy experiment to do. We had better resolution than that, though. We could be able to go, in fact, in every single volume and see, was every chapter there? Was every page there? Was every paragraph there? And so that was the first version of what we did in the Simon Simplex collection. And we, in fact, found children that had some parts of that missing, either a paragraph that might be missing. But eventually, we wanted to get down to the single letter resolution. So out of our 3 billion base pairs, being able to see, could there be one single letter that might have been responsible for that child's autism? And so when we first started out doing this, sequencing was really darn expensive, and so we took a shortcut and we looked at the coding regions of the genome, about 1.5% of the genome, and we went through and we cataloged every single one of those letters in mom, dad, the child with autism, and the sibling without autism, and we picked up some of these single genetic variations. And after doing that, we were able to see that approximately 10% of those children have very clear genetic reasons that are brand new because of one single or two single letters that cause that. Now, the cost of sequencing, luckily, has continued to come down, and in fact, we are now in the process, for any of you who might want to use this information, of actually doing whole genome now sequencing of the Simon Simplex collection. So now not just looking at the 1.5 or 2% where the coding regions are, but looking at all 3 billion base pairs through all of those 2,600 or so individuals within the Simon Simplex collection. And as we're doing it, I'm just going to flip through these, we look like we're coming up right now between both inherited genetic variations as well as new genetic variations. Right now, we can account for about 25% of individuals with autism in terms of identifying a genetic factor. So this is certainly not close to 100%, and I don't think it ever will be, because again, I don't think genes are the only factor in this, but as we're able to dig deeper and deeper, we're able to identify even more of both of these de novo as well as these inherited events, with the idea being that hopefully, again, these will be the anchors we can use to understand the brain and biology, as well as the networks and the synapses that are responsible for some of these behavioral differences. As we're doing this, and at this point we've probably identified about 100 genes that we're pretty certain are involved in a combination of both autism as well as related disorders of intellectual disabilities, neurodevelopmental challenges. In fact, as I'll show you, there's a significant amount of overlap between that. And it's not just as if these genes have been randomly plucked from the 20,000 genes we have. It's actually becoming clearer that there's some method to the madness. There's some order to this. And so there are ways that we look at these networks being able to do basically systems biology approaches to be able to understand which genes are related. And two of the themes that are becoming quite clear is that genes that are right at the synapse, so where one neuron is connecting and talking to another, those are critical genes in this process. And that there's another group of genes that are involved in what we call chromatin modification, as well as gene expression, that are kind of like conductors of the orchestra. They're the ones that are very early in development, actually setting down the plan for how that brain is developing, how those connections are going on, and they actually probably play a very fundamental role in terms of brain development and ultimately function. So as we were doing this, we came back to the question though, what's the gender difference? Can we explain that with any of the genetic factors? And if you actually were to look at the chromosomes, one of the things you'd immediately realize is that one of the gender differences is boys have an X and a Y chromosome and girls have an X and an X chromosome. And so one of the things that, was obvious to ask is, well, boys don't have this backup. Women, we always come prepared, right? We always have a backup plan. So our backup plan is we've got a second X chromosome. The men, they kind of fly by night. They've, got this puny little Y chromosome that unfortunately doesn't have all the backups that girls have. And so one of the questions is that partially responsible? That redundancy in terms of the genes on the X chromosome, does that actually protect females to a certain extent? And the bottom line that I'll give you is this is part of the answer. The X chromosome accounts for about 3% of our genes, but when we look at that, we actually see a disproportionate number of genes on the X chromosome that are responsible for intellectual disabilities. But it's very, very clear that that's not the full answer. In fact, that's probably just a small portion of the answer in terms of relative risk or proportion for males and females in terms of developing autism. So one of the things, as I said, and there are many wonderful investigators, neuropsychologists watching this, is to understand, as I said, just what is normal brain development like for just a girl. I mean, boys versus girls in terms of how they, in cultural, in society, how they learn things, how they actually connect with each other. There are different social dimensions in terms of how children grow up that are gender different. And so part of that is probably that, in a way, girls are protective or boys are susceptible, depending on how you look at this. And it's probably not just all about your genes, but it's also about how we're raising children, how we actually, some of the roles that they play in society, as well as, I'll be honest, it's also some of us as evaluators. I mean, when we look at this process, do we look at the world through, female glasses? Many of us in terms of the field are actually females that are doing this, and maybe some of this is some of our bias in terms of doing these evaluations. So as we're doing this, we also see some differences just in terms of behaviors, and I'm sure many of you see this. Girls tend to be more internalizing as we see some of these behaviors, that where the girls are not necessarily the ones that are going to throw tantrums and be very, sort of outward and have lots of externalizing behaviors and being very disruptive. And so in fact, maybe the girls are having lots of issues, but we're just not even seeing what's going on. They're keeping it inside, they're keeping it bottled up. We're not even necessarily aware of what's going on in terms of what's going on in their head. And so part of this may again be how well we're probing, how well we're understanding, how well we're appreciating what some of these differences are. So as we're doing this, let me just go along quickly. We started looking at the different sort of our top 10 list of what the different genetic causes for autism were within the Simon Simplex collection. And in the early days, one of the things we realized is that there was one particular chromosome, chromosome 16 in particular, that seemed to account for about 1% of all of the kids in the SSC that had autism. And so we said, well, let's take a deep dive. Let's see if we can understand the brain better by focusing in on that particular variation. And so we started a program called the Simon's VIP program. And we did this as what we call a genetics first approach. So let's start with the genes and let's take a deep dive. Let's not say we're going to limit this, for instance, to children with autism, but just anyone who's carrying this, and let's understand better how their brains work, and importantly, how it changes and develops over time to see if there's a way that we can actually intervene in a very positive way. So I have to say we had the luxury of doing this well. So as we did this, we had people who came into study sites, places like Boston Children's Hospital, Baylor, University of Washington, and these lovely families, we tortured them for about 2 or 3 days. So we brought them in and we did all sorts of very detailed neuropsychometric testing. We did imaging. We brought some of them back for another day of functional imaging in addition to structural imaging, MEG, EEG. Even on some of them, we did skin biopsies, which are available to anyone who would like to use them. But we did this in a very detailed way to understand what was going on. And the bottom line, which was somewhat controversial at the beginning of this, is that of all our individuals with the 16p deletion, for instance, only about 25% of them had autism. So even though we got started in studying this in the very first place, because it was the most common genetic cause of autism, in fact, just because you have this, it doesn't go the other way around. It doesn't mean you have autism if you have the 16p deletion. On the other hand, what we did see, and this is just a plot, for instance, of IQ. In this particular study, we had the privilege of being able to also look at the parents who didn't carry the deletion, and also the other siblings in the family that didn't carry the deletion. And if you look at the distribution of IQ, for instance, this was a really high functional group. These were power families, and in part because we tortured them and we made them come in, and so you had to be really motivated to be in this study. So these families had IQs in general of about 110, above average, with 100 being average. And if you looked at the distribution now of our kiddos who had the deletion, for instance, they were shifted about 2 standard deviations to the left. So if you use 70 as a cutoff for being intellectually disabled, certainly most of them were not intellectually disabled. They certainly were shifted from what I would have expected them to be given the family unit that they grew up in. Those other genes, the other environment, they certainly took a hit, if you will, but they didn't necessarily all cross that line or that threshold. And in the same way, when it came to autism, about 25% of them crossed the official threshold to make an autism diagnosis, but still all of them had some sort of features, some sort of dimension, some sort of flavor to what we were seeing. And so we measured that, for instance, by using the social responsiveness scale. And in this particular case, I know this gets a little bit confusing, but over here again are the individuals in red, who are the individuals with the 16p deletion. And then here are their parents, and here are their siblings over here. And again, it's almost exactly the same, about a 2 standard deviation difference in some sort of quantifiable measure of social responsiveness. And so again, they didn't necessarily meet autism criteria, but they clearly had challenges, and challenges specifically within the social dimension. Sorry, this isn't projecting very well, but one of the neat things we could do is not just look at behavior, but we could correlate that with literally what was going on in the brain. And so we saw in a certain minority of individuals, they actually had developmental challenges of the brain. They had polymicrogyria, or in some cases even a Chiari malformation. But one of the things that not everyone had those structural changes that were very specific or localized, but everyone had this following feature. We could actually see the volume of the brain in all of them different from their parents again, in the same sort of quantifiable shift. So the deletion carriers, again, their brain volume overall was larger. Interestingly enough, we could study also the duplications for the same exact region, and they were reciprocal. It was like a mirror image. The brains for duplication carriers were smaller. We could look at the gray matter and the white matter, and we could see that the difference, in fact, was not in the gray matter, but it was in the white matter. And in fact, when you look at this, the actual thickness of the cortex exactly the same, but if you look at the surface area, much greater in the individuals with the deletion. So it was clearly an early developmental process that was going on with this particular gene. And in fact, we can even quantify it by looking at a primary sensory signal. So we can use something, Tim Roberts has used something called the M100. So in other words, you can play a tone, and then you can actually measure in the brain exactly when that tone is being heard, and you can look at the delay for how long it takes for that primary sensory, in this case a ringtone, to be registered in the brain. And in individuals with autism, he knows that you see a delay of about 20 milliseconds. And if you look at the controls here versus our duplication carriers here, you see an even greater difference in that very primary sensory way that the brain is just literally registering sensation. And so you can imagine, for instance, if an individual has this type of challenge just in terms of the way they literally hear or see the world, you can imagine how confusing that can start to be when all of a sudden you get sensory overload, when a lot of things are coming in, and you literally can't process all that information quickly enough to be able to make sense of it. So this is a lot of work that's still going on. We've done functional imaging as well to be able to understand in particular different things about language, hearing language, using language, verbalizing. And this is just the beginning of being able to understand this. But again, I want to put out an unabashed plug for anyone who's interested. All these data are completely freely available through SFARI, SFARI base, and even recontactable in terms of several of these participants. And all of this eventually is going to be to study the neural networks, to be able to understand how signals are coming into the brain, connecting one part of the brain to another, to process all of that, and eventually be able to respond and react to that information. As we've done that, we've actually expanded this. So we had started out using 16p. We now have 39 other additional genes that we're looking at within the Simon's VIP community. As we're doing that, we're trying to do the ones that are as sort of clearly related to autism as possible. And one of my favorites in that regard, I'm not sure where I have it on here, but is a gene called CHD8, which is actually what I would say is very close to the sort of primary features that we think of with autism. And one of the points that I wanted to make by this slide is that some of the genes that you'll see that are related to autism are also some of the same genes that you see with intellectual disability, some of the same genes that you actually see. There's an overlap with schizophrenia, even epilepsy, and in fact that probably is part of the reason why we see a certain fraction of our kiddos who have epilepsy as well. And even, believe it or not, some of these with structural birth defects that we see, congenital heart disease as well as other disorders. And so there's a really very complex interplay in terms of this. The genes that we're seeing that are like this tend to, on average, be the ones for the kids who are at the lower end of the spectrum. It's not 100% in terms of those kiddos, but the ones who have more and more challenges in terms of epilepsy, intellectual disability, those are the ones where we have certainly a greater probability of finding one of these single genes that explains all of those different symptoms that we're having. So within this, our ultimate hope, though, is to be able to get in while the brain is still pliable, while we can be able to actually change how that development is working. And so Ami Klin and others have done beautiful studies in terms of being able to identify, even within the first year of life, based on eye contact, who are the babies who are at risk for doing this. Sometimes a little bit overwhelmed with this. These challenges are actually getting bigger, and in some ways they're becoming different. Many of our kiddos are growing up. They're now young adults, even some of them older adults, and it becomes different in terms of those challenges that we face. And unfortunately, our system, our society, our infrastructure in terms of our educational system and other things, we haven't quite yet filled that gap in terms of being able to support those individuals. And so I think that's going to be one of our next big challenges, is to be able to continue supporting those individuals now as they're growing up. So realizing all of these challenges, one of the things that I think I must have had too much to drink that night when I was thinking about this was that the numbers that we had to be able to study some of these things were just not sufficient. And one of the wonderful things about the Simons Foundation is when we think about a problem, we think about what's the scientific problem. We don't think about, like when we write these NIH grants, what's the $500,000 I can use to solve the problem. We think about what's the problem, and we try and build the resources necessary to solve that problem for the community. And as we started thinking about this, knowing how diverse this condition was, how diverse in terms of etiologies, in terms of age, in terms of capabilities, in terms of challenges, how much diversity there was, you couldn't come up with a study of 1,000 people to be able to study this condition. You needed to think big. And so when I started trying to do some of these power calculations and figuring out how big we needed to be, my minimum estimate was 50,000. And in reality, if you were to, buy me a drink tonight, it would probably be even much larger than that by an order of, probably not an order of magnitude, by at least 100,000 or so. But as we wanted to do this, we realized that this was tough to do. This was tough to be able to get this many people together to be able to address these questions. And in particular, for any one researcher who might be out there, it'd be overwhelming, right? So this was something we needed to be able to do for the community, to be able to make you guys really good at what you do. We need to meet you halfway. And importantly, we wanted the community to partner with everyone to do this. This was going to be a group effort jointly to be able to get this going. The wonderful thing is, as we've thought about doing this, times have changed, right? And so my colleagues from Ian are, I think, stellar representatives of this, about how to be able to use the internet and be able to use social media in new ways that we never thought about before. And so we started thinking and brainstorming with them in terms of how we might be able to do this and how we might be able to put these cohorts together. So as we started thinking about this almost exactly a year ago, one of the things we realized is that we were humbled by the fact that we knew a little bit, but there was sure a whole lot that we did not know. And so the first thing we did is we went out and we sought advice from friends and from families in terms of the ones who were actually facing these challenges, as well as the research community who might be able to use these cohorts. So when we started asking them about this, and we started thinking about what would be of use to them, what would be of use to the research community, we thought one aspect of this might be collecting epidemiological data on exposures, collecting data on time course, and also being able to collect biospecimens to look at the genomic aspect, again, as one portion of this. As we did this and as we talked to the community about things that we might be able to partner with them, and this was kind of a new concept, being able to, they give to the community, or they give to the research community, and the research community also gives back to them. And as we were thinking about how to do that, we asked them, which one of these things actually would you find useful? You know, we don't want to do things that are going to be wasteful with resources. And one of the things that they said is the genetic information, that would be kind of neat. You know, I'd really like to have that. Maybe that's been hard for me to get through my doctor. And so that would be something that might help and guide us in the future. In addition, as you're collecting all of these data about our, you know, little one or about myself, I'd like to see it. I'd like to see how I'm doing. You know, let me see where I am in terms of distribution compared to other folks. Let me get some insight into that. Maybe it'll help me sort of come to realizations about what the particular challenges might be. Cash is always good, and so one thing that was right up there, number 3, was $50. Good incentive in terms of being able to do this, and also being able to be aware of what additional research opportunities were there. I think there were a lot of people who wanted to be able to work with us and do research studies. They just didn't even know what the options were or what was available. So as we did this, we started thinking about this and building all of this into what we were doing. As we also gave them other options, they said, you know what, you researchers, you're really smart and you've got a lot of insight. We'd love to hear from you. We'd love to hear what you're doing. We'd like to hear about what your research is. We'd like to hear your perspectives, whether it's because you're online doing a webinar, because it's a chat room and we can, you know, be able to phone in or type in questions. We'd love to hear about all of those things. We'd also like to be able to understand better what our local services are. You know, there are all sorts of things that you know need in the community on a very practical way. So, you know, if I could have an Angie's List, you know, in terms of for autism in my community, that would be great and that would be really helpful. So as we did this, we listened to the community and we've actually been building this new study to be able to think about all of that in mind. We also, some of you may have been at IMFAR this last year. You'll know that we actually put out a similar sort of survey to researchers. We wanted to hear from your point of view. What are the things that you need? And I'm just unabashedly telling you right now, if you've got any bright ideas, now's the time to tell us. We are all ears and we want to hear from what would be useful for you. And we also actually very specifically targeted our adult population as well. So the individuals on the spectrum who are adults, because we do know that they have very specific challenges and there are going to be very specific needs and interests that they have. And we, again, as I said, want to make sure we're taking everyone under the umbrella as we're doing this. So as we're getting ready to launch this, and I'll tell you that the big launch of this is going to be in April 2016. We're getting ready to start our pilot version of this, or a pilot study, next month. I have to say we're a little bit nervous about this, but we're getting ready to do that soon. And this is actually going to be individuals coming in what I call a volunteer army. But they're coming in and we're going to have them volunteer in a somewhat organized way. So again, one of the big contributions is going to be from our IAN colleagues. There have been many individuals over many years who have joined IAN. We want to give them the option of being able to now expand what they've done with their contribution to IAN and be able to even get more out of it. So it's going to be hopefully a really powerful partnership in that. We've just recently selected 22 clinical sites around the country. We've made them very geographically as well as ethnically diverse to be able to get everyone in the country close to hopefully some center. So there are going to be 22 centers that folks can come through those clinics, and I'm proud to say that one of them is going to be right here in Baltimore. So that's very exciting for us. In addition, we realize that families themselves and local advocacy groups are a powerful partner in terms of doing this. So as I'll show you, we've got many of those. And then we're also running a national media campaign. And so we're going to be able to advertise and take anyone who wants to volunteer and join this effort. As we're doing this, as I said, we know that there are already 20,000 individuals with autism as well as their family members who are in IAN, a very large community, and we're going to be able to give them some of the first opportunities to be able to be in because we need their advice and we need some of their help in terms of figuring out where we probably don't have things quite right, and they're wonderful partners in being able to go forward with this. As I said, this is a map of the United States in terms of our sites all around the country, and you'll see yourselves right here on the map, but we've got all of these hopefully in centers of really fantastic excellence in terms of diagnosticians who really know what autism is. We'll get those individuals into the SPARK campaign as we're doing this, as well as we said, being able to try and get both rural locations as well as big cities, parts in the South, for instance, that haven't necessarily been part of other large cohorts before, but being able to have everyone around the country. And we're going to have materials both in English and Spanish to be able to make sure we can capture capture as many people as we can within the country. As we're doing this, we're very much going to both the large as well as the small state-run advocacy programs. And again, around each one of these 22 sites we have around the country, very specific partners in terms of making sure we've got boots on the ground to be able to think about this and help with us. And as they do this, we're trying to make this as simple as possible for families. We realize that life is crazy, busy, and that really it's not something that people have a lot of time to do, even though they might be quite willing. So this whole process in terms of being able to join the study should take less than half an hour, probably about 15 minutes online to be able to enter your information, kits that'll come straight to your home to be able to donate a biospecimen, a saliva sample in this case, being able to literally spit in a cup and throw it into the mail and be able to send it back for mom, dad, and the individual with autism. And so as we're doing that, we're hopefully making this as simple as possible. You don't even have to get out of your jammies if you don't want to be able to register and do this, because like we said, we know the heart is willing and we just have to enable people to do what they'd like to be able to do. As we're doing this, believe it or not, we realize that one of the challenges for individuals with autism is even spitting is not easy. So we've even thought about creative ways to be able to collect biospecimens. We hate needles. We know kids also in general, but especially kids with autism hate needles. So we've done everything we could to be able to avoid that. We realize that spitting seems kind of gross to certain people. But on the other hand, we know that some kids with autism actually, even in terms of just toothbrushing and oral hygiene, have trouble with that. So we're actually rolling this into something that'll be productive. How you can do this to take care of your teeth in the same way, learning some of those same lessons that you can learn as you're giving your biospecimens. So we're hoping to roll this up into something that's mutually beneficial for everyone involved. And as we're doing this, what we're doing is also developing, hopefully, some powerful resources for families so that this is something that they continuously get new information, new resources, new tips of the day in terms of being able to face the daily challenges. So as we're doing this, we're hoping not to just recruit and then say goodbye to people. Really, we want them in this for the long haul because we want these individuals actually available, shovel ready, ready to do your research studies. We want these individuals hopefully to be available, accessible, both online as well as in person if you have research studies in Baltimore, and that you can, as a researcher, decide, well, maybe I have very specific inclusion criteria. I only want boys who are between the ages of 5 and 7 who have seizures and autism. And we'll be able to just immediately plug up, you'll actually be able to go online and say, these are my criteria. I've got 6,000 kids who meet those criteria, and immediately I'll be able to send out an invitation to them. And if it's something like an online survey, boom, your recruitment done in a matter of a week. But that's what we're trying to do, is produce something that will enable you to ask the questions that are important and do it with minimal burden to you to get the most science done, and being able to partner with the families so that we can advance the field as rapidly as possible. So as we're doing this, one lesson I've learned is that you have to start small so that when you make mistakes, you make small mistakes to start with, and then eventually learn from those mistakes. So we're starting the pilot this fall, this November, in fact, to be certain. As we're doing this, we're going to roll out the entire, as I call, end-to-end, making sure that every different part of this process works. And as we're doing this, we're actually going to be, as I said, returning genetic results specifically to families. And this is kind of a new idea. We're trying to make sure that we're doing it responsibly, that as we return this information, that number one, it's right, number two, that families understand this information, and that they can use it in a positive way to access resources they need. And so this is, I have to tell you, no small order. So we're trying to do this in a small way, as I said, responsibly, to make sure we get it right. And as we do it, we're building this up so that, as I said, by the time of April, and by the time some of you are at IMFAR this spring, we'll be able to tell you how well it works and be able to now start the national recruitment as we go forward. So with this, we're very excited because we think that this will just begin to start providing the resource that many of you need to start addressing some of the big questions. We realize that this is just the beginning, and as we've done this, if it makes you feel any better, we've actually figured out the 10-year budget for this. So we're not thinking that this is going to be just a little tiny thing. It's probably going to take us about 3 years to do the recruitment, and then, as I said, we're thinking about this as a long-term, this is kind of like the Framingham of autism. This is like the women's health study of autism, being able to get these cohorts, hopefully from the time they're little, but also from the time they're big, and just continue to follow them up as they're aging and as they meet different challenges in their lives. So as we're doing this, as I said, we'd love to hear feedback from you, but most importantly, we want you to start those wheels turning. Think about the things that you would like to do if you had access to these folks. Think about the questions that you're going to need answers to, and whisper in our ears, because then we can start collecting the data from the very beginning as we go forward. So as we do this, I just have the privilege of being up here to tell you about this, but it's really a fantastic team that I have the privilege of working with, folks from the Simons VIP study around the country, and then our staff centrally at the Simons Foundation who are working on the autism cohort, and then of course our friends and colleagues at EM. I'll be glad to take any questions.
Questions?
Thanks for sharing this vision. I was wondering what the recent findings about neurons mutating, even in mature neurons, and the extraordinary variability we see in related neurons in adults, what the implications are for thinking then about genetic studies, how they're powered, how they're designed, and does that fit into your thinking at all?
Right. So, so I'll repeat the question, and then I'll also I'll give you my spin on what your question is. So the question is, if I can paraphrase, so there may be mutations that are constantly ongoing in the brain in different neurons, and of course one of the limitations is I'm not poking at people's brains, I'm not sampling that, and so how can you actually get into that? How can you get any insight, and how do you start to tease that apart? So I'll take the very brief sort of way of doing this because it's a really tough and important question, but really hard to be able to answer. So as some of you know, Chris Walsh has started doing this, in particular with certain conditions that were associated with either brain malformations or some forms of epilepsy where we had to do surgery, or one of the initiatives we have is actually post-mortem analysis of the brain net in particular. So those are circumstances when you can actually get access to the relevant tissue, and in fact there are, we would call them either somatic mutations, so things that happen not from the point of conception but as the brain is developing, and could even happen over one's life course. And in fact, as many of you know, that's essentially what cancer is that accumulation of mutations over time. The problem is that we can probably give you, any of us, minimal estimates in terms of how big that burden is, and it's because until we get to the point where we can actually sample every different part of a person's brain, I don't think we're ever really going to know the answer to what you're talking about. We have developed some technologies, Chris Walsh is one of them, there are others, where you can detect very low level of mosaicism in the blood, and if you can essentially sequence deeply enough, you can address the question. But the problem is that many of those mutations will never be in the blood. The only place they will be is within the neurons in the brain. And until we get a way of doing essentially a molecular readout that we can do non-invasively, I think it's going to be a really tough nut to crack. On the other hand, as I said, and again, it's an unabashed plug for this, the Autism BrainNet is the way that we are trying to start addressing that. And I think the more evidence we can get from that, one way or the other, that it's a big burden, the more we'll know in terms of what future resources to put in that particular question. Yes.
Wonderful talk. Thank you very much. You had very interesting information about gender differences. As a clinician, as we know, I've seen sometimes the most severe cases of autism in females, although they are less frequent. They're getting really affected. I'm wondering, did you look into these differences, and did you have any idea, any shed any light?
Sure. So I'll just give you one brief example with a paper that we recently published on a gene called DDX3X. So I talked about the X and the Y chromosome, and in fact I talked about, in some cases women have sort of a backup copy, and maybe that's what protects us. In fact, with some of those genes on the X chromosome, it may be counterintuitive, but in fact we see a deficiency of males with those mutations. And in fact that's because we think in many of those cases the males are actually lethal. The problem is so severe that the males just simply don't make it to term, and they're naturally lost during the pregnancy. So it is possible, I won't say this is the explanation for all of it, but it is possible that some of those very, very severe lesions are in fact in the females due to mutations on the X chromosome, where there's essentially an ascertainment bias in survival for who makes it to birth. So that may be one explanation.
Is there any higher prevalence of autism in children who are preemies or low birth weight?
Very good question. So definitely increased prevalence in terms of premature infants. And so a lot of the efforts that many people in this School of Public Health do in terms of trying to prevent premature birth do great things also in terms of preventing neurodevelopmental disabilities in general and autism specifically. So absolutely.
Yeah. So I'll remind you to repeat the question.
I'm
again,
sorry.
but that's not my question. getting back to the sex differences, so is there thought to how much you might want to oversample female cases?
Very good.
Collection.
So I will repeat the question this time. So the question is, in terms of trying to get at the sex difference issue, how can you start to pick that apart essentially? Do you need to over ascertain females? And in fact, that's been part of a specific request, I should say, for specific researchers. And in fact, if one's only goal is to identify genes, bang for your buck, you'll do better by ascertaining females than you will for males, because as I showed you, there's more enrichment for that. So on the other hand, we don't want to exclude anyone, so we're actually, in the strategy we're using for Spark, not oversampling for females with this first initial pass, but we certainly, in terms of taking very deep dives, will probably look very, very carefully at the females who come in, because from a genetic point of view, because it is likely that they're going to have a higher burden, if you will, of genetics on their side. It's a great question.
All right, let's thank Dr. Chung again.